Good afternoon, everybody. I welcome you all for this presentation on identification of fertility restorers and maintainers in pigeonpea by Mr. Sudhir Kumar. Sudhir is currently pursuing PhD in Bihar Agriculture University, Bhagalpur, Sabur, and he did his master's here and he worked under the guidance of Dr. K.B. Saxena and subsequently under my guidance. And now I invite Mr. Sudhir to present his master's work on identification of fertility restorers and maintainers in pigeonpea. Please, Sudhir.
Thank you, sir. Good afternoon to all of you. Today I am going to present my thesis seminar. The topic is Identification of Fertility Restorers and Maintainers in Pigeonpea. As you know that pigeonpea is important legume crop with a protein content about 20 to 22%, and it represents the first legume where hybrids are available at commercial level. From utility point of view, all the parts of pigeonpea is economically important, so it is also called as 5F crop. In India, it is second most important legume crop and mostly used as dal, and these are measuring in production. Still, India has lower productivity with respect to the world. So, from this data, we can also say that the area and production of pigeonpea is on increasing trend, whereas the productivity is still stagnant. Now, in my observation, there are three objectives. The first objective is to estimate the effectiveness of CMS-lines, to identify the fertility restorers in pigeonpea genotype, to estimate the heterosis. Now, for this purpose, a total 20 hybrids were synthesized by hand pollinating 5 CMS-lines with 11 male lines in kharif 2012 to 13. All these 20 hybrids with 2 check, Maruti and Asha, were shown in randomized block design at ICRISAT. For the observation of data, we take 5 competitive plants for observing these flowing data from days to first flowering, days to 50% flowering, days to maturity, plant height, number of primary branches per plant, number of secondary branches per plant, number of pods per plant, number of seeds per plant, grain yield per plant, 100 seed weight, grain yield per plot, and also for the grain yield in kg per hectare and percent sterility fertility level. From ANOVA, the mean sum of a square, either mean sum of a square for the all these 13 characters are significant, so we can say that all the genotypes are significantly differ. For the first objective, we have taken 5 CMS lines. These all lines are A4 cytoplasm based, and its parent is Cajanus cajanifolius. For the study of pollen sterility level, we utilize 2% acidocarbon solution, and after this we get the result that all these 5 CMS lines has good sterility level, and all these are more than 95%. Simultaneously, we also study the pollen fertility percentage in all these 20 hybrids, including these checks. We find that out of 20, 18 hybrids has more than pollen fertility level 80%, only 2 has below this. For objective third, for the heterosis, heterosis refer to the superiority of F1 over its mid-parent, better parent, standard checks in one or more character. It may be toward the negative or positive with respect to the desired finding. In this research, we mainly focus on standard heterosis, that is economic heterosis. For this purpose, we taken standard heterosis against both the check. SH1 represent the standard heterosis over Maruti and SH2 represent the standard heterosis over the Asha. For this, first flowering to days to maturity, all the hybrids show positive heterosis over the Maruti, but for the Asha, all the hybrids behave like similarly like Asha. For plant head, all the hybrids are taller than both the checks except one hybrid, that is cross ICPA 2048 crossed with ICPA 20093. Number of primary branches with respect to both the checks are in the positive direction, and all the hybrids show positive heterosis for the number of primary branches. For the number of secondary branches, some hybrids are also shown negative in case of secondary branches, but also some are showing significantly positive. For pod per plant, most of the hybrids show positive heterosis for the number of pod per plant with respect to the Maruti, whereas only 4 hybrids are showing positive standard heterosis over the Asha for pod per plant. In respect to seed per pod, all the hybrids show positive heterosis on both the checks. Similarly, for 100 seed weight and yield, most of the hybrids show positive heterosis over both the checks. And for the heterosis for yield, we find that the hybrid hybrids give about against Asha it give about 28% heterosis, whereas against Maruti gives up to the 48% heterosis for the yield. For grain yield, we find that all the hybrids has higher yield with respect to the Maruti, and there are 14 hybrids showing higher yield with respect to the Czech Asha. The highest yielding hybrids is ICPA 2047 crossed with ICPL 20126 that give yield about 26 kg per hectare. Now from this we can conclude that all these 5 CMS lines expressing high level of pollen sterility. Out of 11 male lines, 9 restore fertility more than 80% and may be classified as good restorer parent for corresponding CMS lines, and most of the hybrids show heterosis in desirable direction. Now, I want to thank Dr. Sudhir Kumar sir, Dr. Saxena sir, Dr. PK Singh sir, pigeonpea breeding team, Dr. Rojana Mila and LSU, Housing and Food Service Committee. And also thanks to all those teachers and staffs that may help me in any way for the research. Thank you to all of you.
Yeah, thank you, Mr. Sudhir. Now the hall is open for any questions or queries or clarification. Yeah, just I will ask you one question.
Yes, sir.
So in Bihar, pigeon pea is one of the important crop.
Yes, sir.
So just you highlight what are the production constraints there and what are the building strategies which are to be followed there.
Yes, sir.
To improve or enhance the productivity of pigeon pea in Bihar.
Yes, sir. In Bihar, basically regarding hybrids, first I told about the hybrid. Hybrids are not so effective because we are going to extreme cool in this month of December and January. That is the flowering time for the pigeon pea. So first flowers of flower going drop due to the extreme cool. So this is the study point of view. If any hybrid that have the cold tolerance ability, they can perform good not only in the Bihar, including also some parts of UP. And regarding inbreeds, if any hybrids you taking medium duration or early duration, they are going to longer duration. If you take medium duration, they go to the longer duration due to the temperature fall. Yes, sir.
So we can think of your PhD program in terms of screening the available material for the frost tolerance.
Yes, sir.
Because you will have the frost or low temperatures below 5 degrees in the month of January and February.
Yes, sir.
Then most of the varieties are hybrid shade flowering, and again they initiate flowering by the end of second fortnight of February.
Yes, sir.
So maybe you can have a program for your PhD so that you take all the alert lines and under your conditions you screen for frost tolerance.
Yes, sir.
And subsequently you can develop or you can find out the germplasm lines or improved varieties which are having some sort of frost tolerance.
Yes, sir.
Then you can start off converting those lines into male sterile lines and you can have the hybrids because your soils are highly fertile and expression of hybrids will be much higher. You can harness nearly 40 to 50% of heterosis from hybrids in Bihar compared to even southern India or parts like Madhya Pradesh.
Yes, sir. Yes, sir.
You stated here that out of 11 male lines, 9 restored
Good. Yes, sir. More than 9.
and may be classified as good restorers. What do you mean by may be classified as good restorers? And among these 11 lines, which is the best that you can recommend for pursuing such?
Yes, sir. Sir, because this is the for my observation, this is the 1 year data. So maybe I utilize maybe classified as good restaurant, maybe because it requires more research on this point of view. And these 9 lines are giving more than 80% pollen fertility restoration. That means those corresponding male lines that give more than 80% pollen restoration activity in that particular CMS lines that may be used as good restorers.
So what are these lines already? Can you please flash back your...
Yes, sir.
So where is that line?
ICPA 2047 crossed with ICPL 20126 that give the maximum yield.
No, basically all the hybrids have shown heterosis over Maruti and remaining some hybrids have shown heterosis over Asha.
Yes, sir.
Here basically the problem was you should not have taken Maruti as a check because of the duration. Hybrids and Asha belongs to the same duration of 160 to 180 days.
Yes, sir.
And Maruti comes under 140 to 160 days. No, it's okay, it's fine, the results are fine, but only the thing is standard heterosis you have to compare only with Asha, not with Maruti.
Yes,
I
sir.
have a question. You used the Estocarmine solution, right, to check whether the lines are fertile or sterile, right? Did you see any difference once you planted them on the field compared to your lab results? Like if you test the lines with that solution and say that this is restorer or this is sterile.
Yes, sir.
And when you plant them on field, do you see any variation?
Yes, sir. It is variable because firstly we study about what is the amount of pollen is viable pollen.
Yeah.
So it is often cross-pollinated crop. So there is possibility to cross-pollination. In some land maybe the pollen viability percentage is low, but for setting maybe go. It depends on the availability of a population of bees.
Or it was even at your university location?
Initially this program is also my university, but due to flood they are lost in my location.
You have not mentioned the results of your
Yeah.
university there?
Yeah, ma'am. After that my synopsis is modified at the university level and they are only giving the do here work.
Okay. So I feel overall you have taken the observations.
Yeah.
Almost, how many? 11 trials, 11 trials, right? How many observations have taken? I think 11. What is it? So after this, all these observations, means which is the important trait that is imparting the hybrid vigor? Means you feel that this trait should be considered while considering for then potential hybrids.
Yes, ma'am. From this we can say that Seed per pod is mostly all the hybrids in positive direction on the both the checks. And also for the number of primary branches, mostly most of the hybrids show significantly positive heterosis on the both the checks. I think that play important role.
In all the hybrids?
In most, except 1-2 hybrid, most of the hybrid.
Okay. Thank you.
When you checked those female lines, are they all falling under the same background, right?
Yeah.
You used one background.
Yeah.
Did you have any chance to try other backgrounds with the same potential lines, those restorers or whatever?
Sir, practically this cytoplasm form is feasible for the hybrid production in this time. But another, I don't know.
Being an MSc research, it's
Yes,
going to be only one season, a couple of seasons, right?
Yes, sir.
Yeah. Why I'm asking is some of the lines which show restorer reaction with one background, they may show some other reaction with some other backgrounds. Maintain a restorer. Do you know the gene action also?
No, sir. I don't study it.
Exactly. At that level, it is fine.
Yeah, what's the highest heterosis as compared to your standard check?
Sir, a standard check, best check is Asha. So with respect to Asha, we get maximum heterosis up to the about 28%.
28%. Okay. Now,
Yeah, 48%.
do you
Yeah,
think?
please, if you have any questions.
Okay, do you think this 1 year study is enough, or you should what else you should do?
No enough, sir. Because in hybrids, we study about the location to location is very high variation for the production point of view. So it may be require multiple location and like this.
You see, basically you have collected a lot of data and you have in fact put the objectives only on heterosis. But you might have generated the data for correlation, path analysis, and you can use those for the purpose of publication.
Yes, sir.
Because that is the basic requirement to have some sort of publications from your research work. And also you can publish this particular pollen fertility and restoration studies as well as the aspects of heterosis also. So you analyze all the data for correlation and path matrix and you go for publication. Okay.
Thanks, sir.
Yeah, if there are no questions, thank you very much, Sudhir, and we wish you all the best.
Thanks,
And for your pursuing PhD at BAU, and hope you will work in pigeonpea also for pursuing your PhD degree. And we wish you all the best from the pigeonpea building unit.
Thanks.
Okay, thank you very much.